from 20 to 24 November 2023 on behalf of the students, staff and management of the Vishnu College of Pharmacy. The main objective of the FTP to update you the information about technology-driven pharmaceutical innovation, how the emerging technology helping in pharma innovation, and my sincere thanks to all speakers for acceptance of our invitation. Even though in spite of their busy schedule, I wish you all the best to the faculty and students and make use of the session. Thank you. Over to Bharatam.
So now we request Sujit Kumar sir, Associate Professor, Department of Pharmaceutical Chemistry, to introduce today's speaker, Bharatam sir, to the audience.
Thank you, Bharatam sir. I'm audible?
Yes, sir, you're audible. You're audible.
Thank you. So, taking the pleasure myself to introduce our today's speaker, Professor Prasad V. Bharatam, Department of Medicinal Chemistry, Naipar, Mohali. He has expertise in organic chemistry, medicinal, quantum chemistry, lots of awards and honors to his credits, like fellowship from Indian Academy of Science, Ranbaxy Limited, Research Awards, IBM, Royal Society of Chemistry, London, many more. He has nearly 28 years of teaching experience. He has published various papers in reputed and impact journals. He has also participated in many of his research findings and also contributed to books and book chapters, including patents. He has guided many PG and PhD students. He has received many DST, CSIR, UGC and other funding projects. To his credit, he has various research collaborations with many eminent and professional personalities throughout globally. So, we are very happy to welcome you, sir. And today's topic will be Pharmacoinformatics in anti-malarial drug discovery. Thank you, one and all. Over to Bharatam sir.
With this brief lines of introduction, we request Bharatam to please start the presentation. Over to you, sir. Bharatam sir, your audio is not audible, sir. You are in mute, sir, actually.
Okay, can you see?
Yes, sir, we can see that. We can see you, sir. Your audio is perfect, sir.
Can I start my lecture?
Yes, sir. Yes, please, sir.
Hello.
Yes, sir.
Okay. The topic is about molecular modeling and informatics. I am very happy that Dr. Vishnu College is hosting this event. And I know the college is very progressive and they are doing very proactive steps they are taking to promote pharmaceutical sciences across the campus. And I am very happy to be part of this college. And I am happy to give this lecture. Today, I am going to share my experiences in molecular modeling and informatics in anti-malarial agent design. I hope all of you can hear it and you can see my screen. I would like to tell you that the very first step in any drug discovery process is to perform computer-aided modeling. because the more we use computer approaches, that more time we will be able to save, and we will be able to approach the problem in a rational manner. So I always suggest you to do molecular modeling first, and then organic synthesis, then go to the biology. That is what we do in our lab. To perform organic synthesis, we need a chemical laboratory. To perform computer-aided modeling, we need much less investment. That is, we need a good quality computer and good quality software. Once we have these two, first thing we have to do is we should not start our work with the computer. First, we have to understand the problem. We have to understand the problem thoroughly in the language of chemistry, in the language of biology, in the language of drug discovery. We have to define the problem thoroughly from all possible angles. The moment we succeed in that, then we can start doing the research. So the best approach is to look at the molecules in 3 dimensions. Once you know the molecules in 3 dimensions, you can know the small molecules as well as macromolecules in 3-dimensional arrangement. And once we are confident about these, we can know the properties. Some of the properties are chemistry level properties like nucleophilicity, electrophilicity, hydrogen bonding, van der Waals contact, and other details. Then several other details are at the biological level. What is the IC50 value? What is the MIC value? How the molecule is interacting? Which interaction is hydrophobic in nature? Which interaction is hydrophilic in nature? Okay. Likewise, we will be able to estimate. To do all that, we have some tools available today. Those tools are known as molecular modeling tools. So I am going to show you my slides. Molecular modeling and informatics in anti-malarial agent design. This is the topic. Molecular modeling deals with 3D structure observation. For example, the very first point here is observe the molecule in 3 dimensions. Observe the macromolecule in 3 dimensions. It has got alpha helices. It has got beta sheets. There is hinge region, and there is a substrate binding domain. There is a allosteric binding domain. There is the ATP binding domain. There is a metal binding domain. Then there is a peptide binding domain. There is a covalent bond formation domain. Likewise, inside the protein, there are so many details. Once we understand those details, we should start doing the work. That work can be based on any of the quantum chemical methods, that is structural bioinformatics, structure-based design, or pharmacophore mapping, QSAR, molecular dynamics, molecular docking, quantum chemical methods. Likewise, there are so many techniques that are available. All these techniques have started becoming available after 1994. Before that, these facilities were not there. But also to carry out these facilities, software became freely, widely available only after 2010. So there are several software suppliers in India, and some software is also free. When we should be able to identify, can we invest on purchasing software, or can we use the freely available software from the website? there is small difference. The purchased software is professional in nature. The downloaded software is somewhat old and somewhat not very user friendly. This is the only difference. So we need to take a decision depending on the available depending on the available availability within our institute and depending on the expertise also. Sometimes we are experts in QSAR. Sometimes we are experts in molecular docking. So I need to get the software which is really appropriate for that. Once we do that, we can start doing the work. So this way, what we do is that in our laboratory, we are always working on looking at the 3D structure of small molecules and macromolecules first, and then use these technologies, then design compounds. The new compounds are designed, their 2D structures and 3D structures are understood. After that, we go to the organic laboratory. Once we go to the laboratory, we synthesize the compound, then we go to the in vitro laboratory. After that, we go to the in vivo laboratory. This is the flow of drug discovery process, which is already happening in our institute. And I am going to describe our experience in anti-malarial activity in this particular lecture. So, one of the activities is drug metabolism and toxicity. Every drug discovery scientist should not only know about organic chemistry, he also should know about the drug metabolism as well as toxicity. Because if he is synthesizing a compound which is very efficacious, but if it is toxic, then finally it is meaningless. So a modeling scientist can provide information in drug metabolism as well as in drug toxicity using the interaction with cytochromes. How cytochromes interact with a molecule which I want to synthesize next week. I want to go to organic lab next week. But this week itself, I already know whether this compound will show any toxicity signature that we can find. If it is toxic, we don't want to do the synthesis like that. Okay, this is what we do, and we have published many research articles as shown here in this slide. Next, we also work on drug delivery. Drug delivery also a medicinal chemist should know. Pharmaceutical sciences is an integrated science. Everybody should know each and every topic. Of course, we don't have to learn thoroughly. For example, drug design scientist may not know very thoroughly about drug delivery, but he should have some idea about the drug delivery. Because if the drug is transported through OCT3, we have to prepare it in a different way. We have to prepare appropriate polymorph. We cannot synthesize whatever we want. We have to synthesize a particular polymorph. It should be stable in a particular metastable state. That is why a drug discovery scientist should also know about the drug delivery related information. There is that will give added advantage. So that is what we do in our lab using computer-aided methods. Sometimes we develop software. Small software which are available, big software which are available in the market, anybody can design, anybody can purchase, or anybody can use. But sometimes the software is not complete, and it is not doing the exact job that I want. When it is not doing exact job, I should be able to develop software. So that is what we do in our lab. We have developed a dendrimer builder toolkit. We have developed a predictor 450, descriptor calculator, and then drug likeness tool. Likewise, we have developed some software also. Then we need to go to the specifics about chemistry. What kind of chemical reactions are going on, and what kind of chemical reactions are in progress, and then is it really relevant or not? Sometimes there is a compound with highly electron-rich center, but the highly electron-rich center may not be participating in interact hydrogen bonding. So, do we need hydrogen bond? Is it compulsory, or it is okay in our case? That is required. And then okay, we can first understand chemistry. For example, the 3D structure. Look at the 3D structure of my molecule here. This compound is a divalent N1 compound. This compound has got a CCC angle, CNC angle of 123 degrees. And then this torsional angle is also important. Bond distance is like this. Likewise, I need to understand the 3D details. Then I should look inside the molecule. Inside the molecule means I cannot even see the molecule. How can I look inside the molecule? To look inside the molecule, we have facilities available. These facilities are based on our quantum chemistry. Quantum chemistry gives you molecular orbital pictures. These are all called molecular orbitals. You remember maybe from your electronic structure of nitrogen, electronic structure of N2, whether there is double bond or triple bond. These things we already studied in +2 during our intermediate classes. Now we can do the same thing on entire molecule. When we do that, there is a orbital theory called molecular orbital theory. It will tell us how many sigma orbitals are there, how many pi orbitals are there. So the same thing are shown in pictorial form in contour maps. So this particular you can see that this is called pi orbital, this is sigma orbital. Now, the same pi orbital I can show in this particular contour map, and this is the sigma orbital which is shown in the different contour map. So likewise, we can progress to the presentation. Okay. Now, look at the structure of metformin. Metformin we already know. Most of us are aware of its 2D structure. But do we know about its 3D structure? This is the question. If we know the 3D structure, we know that metformin hydrochloride carries a coordination bond. There is a lone pair of electrons here. There are 2 lone pairs of electrons here in this particular metformin hydrochloride. And then this is cycloguanyl. Cycloguanyl has got 2 coordination bonds and 2 lone pairs. This information becomes very clear when we do our quantum chemical analysis. This work was done and the results are published in these journals from our lab. Now, I will describe how we have taken up this work from the beginning. Somewhere in 1993, we understood the details of imidamide, guanidine, urea, thiourea, selenourea. We started to look inside the molecule. When we start looking inside the molecule, we come to know the electron distribution inside the where is the sigma electrons, where are the pi electrons, how much of overlap is there between these two, how much is the delocalization. All these things we have understood in 1993, 94, like that. These are all the publications obtained from our lab. Next, we have looked at bigonides. Bigonides are metformin, fenformin, then chlorhexidine, all these molecules. Then we looked at the 3D structure as well as the electronic structure. After that, we realized that metformin does not does not look like the structure number 1. Structure number 1 is wrong representation of the metformin, but structure 2 is better. But because metformin is always in hydrochloride state, structure 3 is even better. So 1, 2, 3. If this structure 3 is better than structure 1 and 2, that means structure 3 should be used in molecular docking, it should be used in pharmacophore mapping, it should be used in virtual screening. But structure 1 and 2, if I use, it can lead to some misrepresentation of the data. That kind of information became available to us in 2005. And we published a research paper in Journal of Medicinal Chemistry in 2005. This is a wrong structure. These 2 are right, but 3 is better than 2. That because it has got additional advantage in the form of nitrione character. Having said that, what is the 3D structure? These are the 3D structures. BG1 is better than BG5. Because energy-wise, the energy difference between these two is about 9 kilocalories per mole. That means if I have 100 mole, if I have million molecules of BG1, I have only 1 molecule of the BG5 in my reaction mixture. So I should trust the structure BG1. I don't have to trust the structure BG5. This, how do I know this? By doing a quantum chemical analysis. So, but then we also get information like surface of the molecule. Every drug has got a surface. Every drug has got a surface. This is very similar to our gods. Whenever God Ram's picture is shown, behind the Ram's face, a circle is drawn, white circle, telling that Ram has got this much aura. Similarly, it is true. Every human being has got aura. For example, let us say we are all sitting in a classroom. Our Professor Natarajan sir has entered into our classroom. What we do, we immediately stand up and give him respect, and then we ask him to take a proper seat. Suppose at the same time, after a few minutes, an attendant comes, I give a glass of water to Professor Natarajan. We don't stand up for the attendant. We stand up for Professor, Principal sir, but we don't stand up for the attendant. That is the difference in the surface properties of the two human beings. Similarly, every drug has got a surface property. We have to realize the surface. For example, our metformin has got surface property similar to BG1. If I take the right tautomer, right conformer of my metformin, I will have a structure as shown in BG1. But if I take the wrong conformation, the structural property is different. If I take the wrong conformer BG5, it is very, very different. So you can see that BG1 is better. This is required. BG3 is not important. We should not, we don't want that. BG5 also we don't want. BGP1 also we don't want. BGA2 also we don't want. But we want BG1. This kind of information becomes available to us when we do something called molecular electrostatic potential analysis. Okay, so that kind of confirmation will be complementary with the micromolecules. Such complementarity is going to give us complete clarity on drug discovery. Whenever I want to design a new compound, I want a compound which is similar to BG1, but I don't want a compound similar to BG3 and BG5. That means now I know what I want, what I don't want. Before I spend all my money, before I spend all my energy, I will be able to analyze the compounds in this way. This kind of procedure is known as molecular electrostatic potential, and that can be done very easily using computers. Now, this is what we have done. We have tried to understand the surface property, internal property, as well as the 3D structure of cycloguanil. Why we know that cycloguanil is a drug, proguanil is a prodrug. Proguanil gets converted into cycloguanil with the help of CYP2C19. That information we already have. Because of that knowledge, but that is biology knowledge. That is, doctors and biology people have said that proguanil is getting converted into cycloguanil under in vivo condition. Okay, that is excellent news. But then, what is the structure of proguanil? What is the structure of cycloguanil? How this is happening? If I want to understand, we have to see it in 3 dimensions. When we see it in 3 dimensions, it becomes clear that the structure need to be slightly modified. And when we do that modification, we come to know that cycloguanil is very similar to this divalent N1 compounds, which are known as nitriones. How that chemistry came into picture? That chemistry came because of the 1961 research article by Ramirez, which is showing that compounds can be carbones. Our nitriones are similar to carbones. They have this kind of molecular orbital analysis. This is sigma type, this is the pi type. These details are compared. Then I can design many more molecules which can carry similar character. Such compounds are nitriones. That means we have got a clue based on the 3D structure, based on the internal molecular orbital structure, and based on the surface property, we are able to design new class of compounds. These are the new class of compounds we have tried, and then after understanding them better, we have realized that these compounds carry 2 lone pairs at the nitrogen center. These compounds carry coordination bonds. The compounds can be represented in this way in 3 dimensions. And then these are the various details of their geometry. They are responsible for intramolecular hydrogen bond breaking, and conjugation also breaks down. 2 lone pairs get localized at the central nitrogen. A lean-like character is induced, and the dynamism increases through N inversion as well as the C-N rotational barrier. These are all the details that became available. That means the properties of the molecules became available. Just like our properties of our own friends. For example, our friend likes cricket. So that means during the World Cup match, he will not attend any scientific meeting. That is known to us because he likes cricket. Suppose our friend likes to go to temple. That means he will go to the temple of goddess Maulamma in Bhimavaram. So he may be every Friday he goes to the temple because he likes to pray to gods. So we know the properties of our friends. Similarly, we know the properties of our drug molecules when we look inside their 3D structure and their surface properties. This is the first step in molecular modeling. This is all the work done, and then we have designed several compounds. And then once you do design several compounds, we have to go to understand their reactions. What kind of reactions our molecules will undergo? What is the structure is important. Structure is not sufficient. Reactions are also important. How the reaction will progress. Okay, if the reaction is progressing under nucleophilic conditions, or the reaction is progressing under oxidizing conditions, or the reaction progresses under the hydrophobic conditions, there are many details. We can also know not only from the organic chemist, from the computational chemist also. By doing computer-aided modeling, we will be able to know. For that, we have facilities called molecular docking available. If suppose my molecule is interacting with cytochromes, that means it will undergo oxidation very well. Suppose my compound is interacting with PFDHFR, that means it may be showing the inhibitory activity. If my compound is showing interaction with PPR gamma, that means my compound is an agonist. Sometimes my compound may be antagonist. I can know the details by performing the molecular docking analysis. So before that, we have understood how the oxidation will happen. This is the reaction profile. And then for this reaction, for the first step of the reaction, it will lose 1 kilocalorie. But the barrier is 16 kilocalories. But on the other hand, this particular next step of the reaction requires releases an energy of 45 kilocalories per mole. And then the cyclization will happen very easily. Cyclization does not require more than 2 kilocalories. But the formation of the triazotrein releases 45 kilocalories, but the barrier is 16 kilocalories. So once I know this much, I will be able to take a decision. For example, I want to go to market today. If I know that at this time the traffic is very high, or a train is passing through the railway track, it is not good time. Too many trains to go this time. Better go after 1 hour. So if I know that detail, I will be saving my time and my energy. Similarly, my reactions, my energy profile of my chemical reactions tells us whether to synthesize the compound or we should not synthesize the compound. Because if it is too costly, our principals may not give the funds. If it is so, you can suppose you tell that, sir, this is very easy to do. You can do this reaction within 1,000 rupees. So principal sir will immediately sanction 1,000 rupees to us while doing to perform the reaction. Okay, if you ask him, sir, we need 1 lakh rupees to perform one reaction, sir will have difficulty in sanctioning that much money. Right? So we need to tell him, and before requesting some funds from our sir, we need to tell him what is the mechanism, what is the process, how much money is required, how much easy it is to do, what you will be able to do within 1 week, what you will be able to do within coming 1 year. You need to be able to promise to him, and then he will get the confidence in us. Once he gets confidence, he will give us funds to perform the reactions. To get this confidence, our computers will help. Computer-aided design, molecular modeling will immediately help in doing this kind of work. So we have gone through several details. There are compounds with N3 plus which can accept electron density from the ligands like this. That kind of work was reported in England Chemie in 2013. And this kind of compounds like famotidine, brutidine here, fungicidal activity is shown by this kind of moiety. A neuroprotective moiety, neuroprotective ability was shown by this kind of moiety. Likewise, there are several compounds called thiazolguanidines. That means thiazole unit is there in our compound. Guanidine moiety is there. These two are combined. These are known as guanyl thiourea derivatives. So this was the first design that was done. So are there anything more we can do? Yes, we can design compounds like this. But guanyl thiourea are docking better. So how to synthesize them? So how to do that? To do so, what we go to the available literature. This is already known how to design this, how to design this, how to design this. So maybe I can use the same tricks. For example, how to design this? You start with a thione derivative and you treat it with potassium or sodium alloy, and then reflects the reaction. Then we will get this kind of carbene. This carbene can be reacted to give this chloro derivative and this amine derivative. Once we react between these two, I will get this compound. So this is the available literature. So we have not done. This is the work reported in the literature. Okay. Now that we know the literature, we studied, And now we can show, yes, this is the compound that we are designing. What are all the features? What are all the pharmacophoric features? One is that this kind of head group is required. The guanidine, guanyl thiourea. This is compulsory in already designed in all the compounds designed by us. There is a CH2 group is required for linker. Linker side chain may have one or two or three CH2 groups. Or R group should be lipophilic in nature. So how to synthesize such compounds? How to do that? This is one of the ways. This is primary alcohol. You start with primary alcohol, treat it with bromine, you will get bromo derivative. This bromo derivative can be combined with this guanidinium compound, guanyl thiourea compound. Then what happens? HBr will go away, bromine from here, H from here, the HBr will go away, and then carbon sulfur bond will be generated. This compound with the carbon sulfur, newly created carbon sulfur bond is nothing but our guanyl thiourea derivative. So we have to make strategies how to design our compounds. That means we have to do homework on molecular modeling, and then we have to do homework on the computational on the chemical reaction modeling. Once we do that, we can go to the synthetic schemes. How to synthesize another scheme. Look at this. Look at this reaction. You start with two bromine derivative, two bromines on two sides, compound number 16. We can condense with guanyl thiourea. We can get this compound with two head groups. Sometimes two head groups are important. Sometimes one head group is important. We can synthesize many molecules in this kind of chemistry. First, suppose you start with this alcohol. This is nothing but a phenol. You react with this bromine. HBr will go away. This kind of linker chain will be formed. Then you take this compound 22 and react with this kind of guanyl thiourea, SC bond will be formed, and this is nothing but the final compound that is desired. So once we do that, we can design many molecules, know their chemistry, and know their how to synthesize them better, how to improve the synthesis procedure, how to improve the yield, how to make it cheaper. That means we don't want to spend too much time, too much money. For example, if to perform to get 1 gram of my molecule, if I have to spend 1,000 rupees, then the cost of the drug will be very high. So I need to know how to synthesize this compound in a much cheaper manner. That means it should be reaching the patient. Poor patient also should be able to buy, and the rich patient also should be able to buy. Our compound will not distinguish poor or rich, but the pocket, how much the patient can afford, will be able to decide whether my compound will be useful or not. If the cost of my compound is like the cost of in remdesivir, for example, do you remember remdesivir? What happened? People started selling in black market for one tablet 20,000 rupees. So that is too much. COVID, how can you control COVID like that? Government cannot control like that. Government found that such remdesivir is useless. We need to find a vaccine. They have done that, and then they have given approvals. So government has facilitated the control of corona. Likewise, we need to make some plans early on to make very cheap and best molecules. This is one method we have adopted. To start with this chloro derivative, condense with this cyano derivative, you will get this CN bond formed compound. This compound can be reacted at 45 degrees methyl iodide. Then you will get this kind of an imine. Once this kind of imine is formed, you treat it with P2S5. Then you will get this kind of an imine where cyano group is replaced with this thioamide group. Once this thioamide group is available, you can perform further alkylation. Then this sulfur will become alkylated, entire molecule will become a cationic species, and such cationic species, when you treat with ammonium acetate, you will get this compound with two NH2 groups. So this compound is now readily carrying the lone carbon-nitrogen coordination bond and a lone pair of electron density here. Two lone pairs, two coordination bonds, and then the molecule is positively charged. So that kind of molecules are required. That has become clear from the molecular modeling. And now that is possible to synthesize such compounds. It has become clear from the organic synthetic procedure. Now we are ready to take up the research in vitro. Before going there, we can get the 3D structure of the molecules. This is the crystal structure of the molecule with all the details. Geometrical details. Now these are the electronic structure details. That is sigma orbital. This is the pi type of orbital. The sigma is visible in this way, and pi is having equal distribution above and below the plane. So likewise, we will get the internal details. Then we synthesize more compounds. Then this is the procedure to synthesize the more compounds. You start with the thiol derivative, convert into methyl thiol, and then give this particular salt with PF6. Then you can convert this. After reaction with hydrazone hydrate, we can get to this imine with NH2 substituted. This NH2 substituted imine, and this compound can be dimerized to give this compound. That is a carb. This is known as an agene, because carbon nitrogen double bond, nitrogen nitrogen single bond, and nitrogen carbon double bond. This is a requirement to call these compounds as agenes. These agenes can be easily designed, and then we can perform the characterization. Characterization is that it has got two lone pairs of electrons at the central nitrogen. It has got coordination interactions. It has got excess electron density at the nitrogen center. Still, this molecule is unreactive. Though it has got excess electron density, it can show hydrogen bonding, but it cannot show reactivity at the nitrogen center. This is because the lone pairs of electrons are tightly bound to the nitrogen center. This nitrogen center is not important, but the carbon center is more important for this. How do we know that? If we look at the 3D structure, we can know that. So it can be important in medicinal chemistry, it can be important in catalysis, it can be important in group activity. Likewise, there are so many advantages we want to exploit. Now what to do? After that, we have to dock the molecule. When we dock the molecule, we will come to know whether it will enter into the cavity properly or not. Because the drug has to enter into the cavity first before it can show drug action. So there is an entry channel, there is an exit channel. Is it the exit channel and entry channel, are they properly working? For example, the insulin receptor. Insulin receptor, if the insulin is present and the insulin receptor is not recognizing insulin, then what is that known as? That is a period of insulin resistance. That means that is the period metformin will help. That is the period where rosiglitazone will help. That is the period it is possible to develop a tablet, maybe 10 tablets, 10 rupees, or maybe 10 tablets, only 1 rupee. We can develop that kind of molecules. So this is a procedure which for that the molecular dynamics is quite useful. How molecular dynamics do you use it? Can you tell me? Any of you know? Maybe some of you know, and some of you don't know. So to do this quantum chemistry, Gaussian software is available. To perform molecular docking, Autodock is available. Autodock Vina is available. Then to perform the same molecular docking, we have a Glide software is available. So likewise, several varieties of software are available. These results are obtained from Glide software. What Glide software is telling that there is a lone pair of electron at this nitrogen center. It is not participating in any bonding. In any hydrogen bonding also it is not participating, but it is there. But why it is there? What it is doing inside the cavity of the PMDHFR? This is a question. So we have to ask questions. If the cyclocomunal is working as a drug, why it is working as a drug is an important question. Because if I know that, I can develop a new compound which is looking very similar to the cycloguanil, but at the same time it is able to inhibit the action of PFDHFR. What is the action of PFDHFR? It is a hydrate transfer. Between what and what? It is a hydrate transfer between NADPH and the trihydrofolate. When that reaction is happening very fast, I can develop more maybe. So we have performed docking of the cycloguanil inside the cavity of the macromolecule. We have performed docking inside the cavity of the PFDHFR. Molecular docking has been done. Then we come to know where is the hydrogen bond, where is hydrophobic interaction, where is the electron deficient center, which is going to accept electron density from the hydride ion. And likewise, we can get many details. Then what becomes clear is that this particular nitrogen center is not, this particular central nitrogen is not doing any reaction, because it can only sit inside the cavity. It cannot participate in reaction, because whatever reactions that it can go, it is already undergone, and all the nucleophilicity and electrophilicity factors are already satisfied. Though this molecule is positively charged, it is not very active. Okay? That is good news. That is good news, because if it is very, very active, it is not possible to control the activity. If it is having the very controlled activity, it is good. Suppose in our class there is one student who is very, very active. He is interested in music, he is interested in dances, he is interested in drama, he is interested in movies. If somebody is interested in all of them, is he interested in my lesson or not? Why principal sir is giving salary to me? He is giving salary to me for me to teach. If my students are all very active and if they are not listening to my lecture, then what is the use of their activity? Okay, so we don't want too much active and we don't want too little active in our drug molecules. So that is exactly what is going to help. That kind of clarity will get through molecular docking and pharmacophore mapping and other technologies. Now we know that it is happening. Why don't I design better, much bigger compounds like P218? It is a molecule in clinical trials. This compound is developed in Bangkok, in University, Mahidol University. Okay, take that compound and try to understand better. One more. Whatever is known information, we have to understand very clearly at 3D structures. So we have done that. After doing that, we have found out molecular docking can be done. when we do molecular docking, we get the result in the form of the binding free energy. Binding free energy tells that this molecule has got a binding free energy of 22 kilocalories. 22 kilocalories are only 22 kilocalories. In this particular example, it is not that it is a good value, it is a very small value. If it is very small value, is it good news for us or bad news? Sometimes it is some good, sometimes it is bad. Now look at the next column here. This value is minus 22.14. This value is minus 34.9. That means my compound is active against wild type PFDHFR, providing minus 34 kilocalories per mole energy release. Whereas this compound, again, in quadrupole moment, it is giving is minus 34. In W mutant, it is giving minus 29. And again, quadrupole kind of thing, it is giving minus 34. So what is happening that my compound is showing interactions with PFDHFR strongly, but it is not interacting with human DHFR strongly. That means if I can develop a tool for this, I can attack the parasite only. Because even if it is active in my, even if this molecule is active in according to docking result, minus 22 is good enough, but it is not sufficiently selective. So no, it is sufficiently selective because it will not disturb the human DHFR, it will disturb only the PFDHFR. That is why it can become a drug. So P218 molecule can become a selective inhibitor of PFDHFR. That is the information that comes out of the molecular dynamics analysis, and that information has been utilized by us to design many more new molecules. Okay, next step is to find out which amino acid is important. It has become clear that arginine 70 is more important than valine 115 because this particular energy values with arginine, my molecule is releasing 70 kilocalories energy, but what is it? 18 kilocalories of energy, but with valine, it is giving only 2-3 kilocalories energy release. So arginine is more important amino acid, and then cysteine is also more important. We need to pay attention to these two. 1, 2, and 3. 3, cysteine 59 and arginine 59 are really very useful amino acids for the selective design of the new drug molecules. That is a clue. Now I can synthesize. I can go to the biology. This is in vitro biology. E. coli, using E. coli biotechnology was done. Recombinant PFDHFR was generated. The PFDHFR activity was determined using spectrochemical methods by measuring the absorbance of NADPH. If NADPH is present, it is 340 nanomolar, a specific NMR peak will be observed. So based on that, we can estimate. So protein content, how much NADPH is utilized, When my molecule is used, how much NADPH is utilized, and when lead molecule is given, how much NADPH is utilized. From that, I can estimate and find out that our compound can indeed bind to PFDHFR with a good IC50 value at about 25 degrees, and that is good news. We keep that information on one side. Because that is good news, we go to in vivo biology. What in vivo biology is telling? The compound can be anti-malarial. That means I have to inject. I have to inject my molecules, not my molecules. I have to inject my rat or mouse. I have to inflict malaria in these mice, and then I should be able to treat the mice. This is the purpose. So what we have done is that we synthesized about 23 compounds. Each of these compounds was converted into 3 concentrations. We have taken about 100 mice and then injected these 23 compounds. And then we found that few compounds are we first injected them with malaria. Plasmodium berghi was given to the normal mouse. Then these mouse react after 4 days. They developed the malaria, and then we supplied the compounds. When we supplied the compounds, the malaria was treated. In the case of 23 compounds, malaria was not treated. In the case of the 6 compounds, malaria was treated out of the 23. That means we are able to identify at least 6 compounds which can treat malaria in the mouse. But we observe the biology, in vivo biology, we have to observe for long periods. We have observed after 14 days. And then we observed after up to 60 days, the mouse. Then we observed that the mice survived happily in case of only 1 compound. In other 5 compounds, malaria was recurred. That means because of the recurrence of the malaria, we cannot continue with 5 compounds. But because the 1 compound is able to treat malaria, as well as the recurrence of the malaria did not happen, so that single compound can be suggested as a new lead molecule for Baker to do synthesizing many new compounds. So likewise, what we have done is that we have done 3D QSAR, we have done pharmacophore mapping, we have done molecular dynamics, we have done molecular docking, and then finally we are able to say that these are the 2 compounds which can show activity at 0.4 micromolar. A second compound shows activity at 100 micromolar, and then use this data to design many more molecules. That's what we have done, and some of these results are published in these journals. So what is it we have done? We found a chemical bonding, that is the covalent bond versus coordination bond analysis we have done. And then we have found that these nitrione kind of compounds are different from others. Out of these nitrione compounds, we can show that some of them can be active, very similar to bicarbonate. Then which are those compounds? Guanyl thiourea derivatives. Then because of the intermolecular interactions, load pair interactions, all of them are very much comparable in this between the cycloguanyl and our newly designed compound, and thus we can go ahead and do a lot more analysis. Still, work is in pending. So I would like to state that we are doing this work that is anti-malarial drug discovery using the computer-aided methods. After doing computer analysis, we go to organic lab. From there, we go to in vitro lab, and from there, we go to in vivo lab. And finally, we are able to declare that there is a this kind of this kind of publications can be obtained, and then patents can be obtained on these compounds. And it is this kind of approach will give us new lead molecules and then many new clues for drug discovery. So with this, I will stop my lecture. I thank you all. These are my teachers who helped me in teaching from Bangalore, from Amsterdam. They helped me. They taught me a lot. These are the funding agencies. I received funding from NIPER, DST, DBT, CSR, GNDU, Punjab University. And then these are the companies which also recognized our work. IBM, Eli Lilly, Johnson Johnson, Ranbaxy, all of them are using our work and recognizing our, appreciating our work. And then these are the students who have done the work, and they are all the top leading scientists across the country and across the world today. Okay. Now I will stop. This is NIPER Mohali. You are welcome to NIPER Mohali whenever you have opportunity. And this is my team, which is working hard. Thank you very much.
Thank you so much for the talk, sir. So now the session is open for questions. So any participant, you can unmute and you can ask any questions you have, or you can put in your chat box. We'll bring that to this. Any questions? Please unmute and. For the participants watching on the YouTube, you can just post the question in the chat. We'll take that and we'll represent that to the sir.
Bharatam sir, I got few questions from YouTube.
Okay.
So I got a question like how to identify the active metabolite of a particular molecule by software.
Yes, there are some predictive software that can give us which is the site of metabolism. So what we do is that we look at the molecule at the electronic level using quantum chemical approach, and we find out where is the electron deficiency, which center is ready to react. This entire process is known as identifying the site of metabolism, SOM. We can do quantum chemical analysis, we can do molecular docking, and we can do predictive tool utilization. And finally, we can find out this particular center in the molecule is more prone to metabolism. Then we can see what kind of reaction can happen. Is it the oxidation reaction? Is it the N dealkylation reaction or O dealkylation reaction? Or what is the reaction that can happen there? And then based on that, we can predict the metabolite. If it is oxidation, we will give you the oxidizer product. If it is N dealkylation, we will provide the N dealkylation product. Likewise, we can do lots of computational analysis. The science behind it is very much involved, but the technology is very useful. We can use it quickly.
Thank you, sir. I have one more question with me. What is the relation between the charges, relation or differences between the charges of a particular molecule and a particular receptor?
Right. If I understand the question correctly, what a particular property is required for the drug action, right?
Is want to know the charge. Is there any charge difference between a molecule and the receptor where it is binding?
Right. To do so, we have to look at the 3D structure of the macromolecule. To do so, we have software called PyMol. We can download PyMol, or we can use Mastro Schrodinger software. We can purchase it, and we can prepare the 3D structure of the molecule and look at the 3D structure very carefully. Once we look at the 3D structure, we may not be able to find the cavity because 3D structure is much complicated. With our own eye, we may not be able to do. So for that site identification, there is a small tool called site identification. That site identification software will tell where is the maximum cavity available. What is the character of that cavity? Is it positively charged or negatively charged? Is it going to show the hydrogen bonding interaction or hydrophobic interaction? Is it going to facilitate the hydrogen transfer or hydrate transfer, or will prevent the hydrate transfer? In our case, we need the prevention of hydrate transfer, and that is why our compounds, we need only inhibitor. We don't want any reaction to take place. So that kind of chemistry we understand, and then we try to give answers to the questions that you are asking.
Thank you, sir. Bapaya sir, I am done with the questions. Over to you.
Questions from any more participants?
Is it clear?
Any more questions from any other participants, please? It is not.
Hello.
Thank you, sir. Professor Bharatam, sir, you are enlightened our faculty and as well as students for today's session. Thank you. And my personal request is that whenever you come to your native place, Bharatam, please visit SVCP and inform me the date, sir, so that I will take care.
So it is not we take. We propose our special thanks to Bharatam sir, who have spent a lot of time with us and enlightening us with his knowledge. And definitely, we are always gratitude. We have gratitude for whatever you have told to us in today's class. So it is not. I also thank all the participants, and we have shared the meeting link. Sorry, feedback link for today's session. Please provide feedback, and tomorrow's session will start at 11. That's all for today's meeting.
Okay, thank you very much. Very nice talking to your students and participating in this event. Bye.
Bye, sir. Bye, sir.
Thank you, sir. Thank you, sir. Thank you, sir.